Hello guys, welcome back to my channel. Today I will be talking about microbial nutrition and culture media and essential guide. To begin with, what is a microbial nutrition? What does it mean? Microbial nutrition refers to the study of how microorganisms obtain the energy and nutrients they need to live, grow, and reproduce. Like all living organisms, microbes require certain chemical substance from their environment to carry out life processes such as metabolism, energy production, and cellular repair. Also, there are two types of nutrients which is required by microorganism. One is macronutrients and that is micronutrients. We will be talking about those also in this ppt. So beginning with the first slide, understanding the microbial introduction to microbial nutrition, why it matters. Understanding microbial nutrition is fundamental to microbiology. Microorganisms from bacteria to fungi require specific nutrients to grow, reproduce, and carry out their metabolic activity. Without a precise nutrition environment, the study, isolation, and cultivation become challenging, if not possible. Research and discovery enables the study of microbial physiology and genetics, industrial application, crucial for fermentation, antibiotic production, and waste treatment. Medical diagnosis essential for isolating and identifying pathogenic microbes from clinical samples. These guide explore the diverse nutrition needs of microbes and the various culture media designed to meet them, providing a comprehensive overview for student and professional alike. So, in simpler words, micro microbial nutrients are the nutrition which is required by microorganism in order to grow, live, and reproduce, also to perform many processes like metabolism, energy production, and cellular repair. Basic nutrition requirement of microorganisms. As I said, there are two types of nutrients that is required by microorganism. One is macro, and the other one is micro. Macronutrients are usually required in larger amount, whereas micronutrients are required in smaller amounts. But macronutrients involve carbon, nitrogen, hydrogen, phosphorus, and sulfur, and whereas micronutrients, also known as trace elements, have includes iron, zinc, copper, manganese, typically act as an enzyme cofactor. I will be talking about macronutrients in the next slides. Over here, let's talk about the macronutrients. All living organisms, including microbes, require a source of energy and raw materials to synthesize cellular components. These requirements can be broadly characterized into several key areas. Microbe exists as a remarkable diversity in how they obtain these necessary, leading to various nutritional classification. Carbon source for structure, molecules, energy, nitrogen source for protein, nucleic acid, and amino acid, energy source to fuel metabolic processes, inorganic salts essential for enzyme function and osmotic balance, growth factors, organic compounds that cannot be synthesized by the microbe. Understanding these fundamental needs to be the need is the first step in formulating effective culture media for microbial cultivation. That's true. Also, we need to know that what is the also we need to know the importance of microbial nutrition. Microbial nutrition is essential for growth and survival. Proper nutrition is essential for microbial cell division, metabolism, and adaptation for new environment. Also, it has a contribution in industrial microbiology, used to optimize condition for fermentation, in producing antibiotics, alcohol, enzymes, helps in designing nutrient media for culturing microbes in labs. Then also it has a contribution in medical microbiology, where understanding microbial nutrition helps in controlling pathogenic microbes, limiting iron to inhibit bacterial growth. Agriculture soil microbes help in nutrient fixation and nutrient cycle, which supports plant growth. Environmental application in bioremediation, microbes are provided with specific nutrients to degrade pollutants effectively. Biotechnology, tailoring microbial nutrition is key in genetic engineering, such as producing insulin and other recombinant protein. So the conclusion of microbial nutrition is fundamental to understanding how microbes function in natural and artificial environments. It has broad application in health, industry, agriculture, environmental management. Let me know if you need. In conclusion, is the microbial nutrition is fundamental to for understanding how microbes function in natural and artificial environment. It has broad application in health, industry, agriculture, and environmental management. So, guys, in simpler words, Nutrition, microbial nutrition is required and has contribution in various fields. Without this nutrition, any process won't be performed in a particular manner, or it won't be even initiated in a particular manner. So microbial nutrition is very much essential, far much than our imagination. So macronutrients are the elements required in relatively large quantity for microbial growth and metabolism. Elements from these elements form the backbone of cellular structure and energy carrying molecules. Their availability directly impact the vigor and yield of microbial culture. As I said, there's carbon, there's nitrogen, there's phosphorus, there's sulfur. Moving on to the next slide. Micronutrients, and also known as trace elements, catalyzers for growth. While required in smaller amounts, micronutrients and trace elements are indispensable for microbial life. They often act as a cofactor for enzyme, enabling crucial metabolic reaction. Their absence can severely impair growth and metabolic function, even if macronutrients are abundant. Iron, zinc, copper, manganese. There is What are the examples of micronutrients? It is iron, zinc, copper, manganese, cobalt, and molybdenum. And each of these has its own function, has its own contribution. The iron is used in electron transport. Cytochromes enzyme cofactor. Zinc is used as cofactor for DNA polymerase and alcohol dehydrogenase. Then copper is used for electron transport enzyme system, cytochrome oxidase. Then manganese is used for enzyme activation, and cobalt essential components of vitamin B, C, and molybdenum is nitrogen fixation, nitrogen enzyme. These elements are typically present as impurities in other media components, or can be added in trace amounts to ensure optimal microbial performance. Then understanding different type of media. What is the culture media? First of all, culture media are nutrient rich substance or solution used in laboratories to grow, multiply, isolate, or identify microbes such as bacteria, fungi, and algae under controlled conditions. So guys, my culture media is not a rocket science. It's just a liquid, or it is just available in a liquid, solid, or semi-solid form, and it has a very basic component. So culture media is not a rocket science. We just need to understand what are the basic components of culture media. The basic components of culture media is water, carbon source means usually sugar like glucose, then nitrogen source which is known as peptones or meat extract or ammonium salts. Then there is minerals. For minerals we use phosphate, magnesium. Then there's growth factor, we use vitamins, amino acid. Then in agar we use a gelatin like substance from seaweed used to solidify the medium. So culture media especially formulated mixtures that support microbial growth. The composition varies widely depending on the special nutrition requirement of the target microorganism and the purpose of cultivation. Choosing correct medium is crucial for successful microbial growth. So there are two types of media: defined, which is also known as synthetic media. Then there is undefined, which is known as complex media. Defined media, all components and their exact concentration are known, and undefined complex media contains extract or edges, so that composition is unknown. Over here, beyond these basic categories, media can be further classified as selective differentiation and enrichment on anaerobic, each with a unique application in microbiology. So, for example, types of culture media. Culture media can be classified based on state. There's three state of culture media, which is liquid, solid, and semi-solid. Liquid has no agar used to grow larger amount of microbes, and solid contains 1.5 to 2% sugar used for colony isolation. Then semi-solid contains less than 1% agar used to check motility or oxygen requirement. Then there is also chemical composition for synthetic and complex media. For synthetic media, the chemical composition is sorry. For the chemical composition for defined synthetic media is the exact chemical composition of defined synthetic media is known. And for complex undefined media, it contains unknown components like yeast extract and peptone. So the most common example of defined synthetic media is glucose, salts, broth. And then there is, for the most common example for complex media is nutrient broth, tryptic, soy, agar. Moving on to the next slide. Preparing and sterilizing culture media for optimal growth. Accurate preparation and rigorous sterilization are paramount to prevent contamination and ensure consistent uh, re- uh, consistent reliable experiment results. Any contamination introduced during preparation or inadequate sterilization can lead to erroneous observation and wasted resources, which is correct. Preparation steps include weighing, accurately weighing all drying ingredients, dissolve ingredients in distilled or deionized water, pH adjustment, adjust pH to the desired range using acid or bases, dispensing, distribute into appropriate containers, test tube, flask. Sterilization is most commonly achieved using an autoclave, which employs high pressure steam, 121 degrees Celsius for 15 to 20 minutes to eliminate all living organisms, including spores. A septic technique must be maintained throughout the process. Selective and differential media, there's various types, there's various type of media. There's enriched media, there is selective media, there is differential media, there is transport media, there is anaerobic media. But in this ppt we will be talking about selective and differential media. These specialized media are invaluable tools for microbiologists, allowing them to isolate specific microorganism from mixed population and to distinguish between different types of microbes based on their metabolic characteristics. So there is selective media, there is differential media. Selective media contains inhibitors that suppress the growth of unwanted microorganism while permitting the growth of the target organism, ideal for isolating pathogen from sample with high normal flora. Include differential media include indicators that produce visible change in response to metabolic activity of certain microorganisms, allowing differentiation. Example include Meconium agar, which is broad selective for gram-negative bacteria and differential for lactose fermentation, and mannitol salt agar selective for staphylococci and differential for metal fermentation. So in easier word, what is happening in a selective media is selective media inhibits some microbes, allow others to grow. The most common example of a selective media is Meconium agar and mannitol salt agar. And what is happening in differential media is it shows visible difference between organisms. Okay, and the most common example of differential media is Meconium agar, lactose fermenters turn pink, also blood agar, which is known as hemolysis. Moving on to the next slide. Anaerobic and enrichment media, cultivating specific microbes. Some microorganisms have unique atmospheric or nutrition requirement that necessitate specialized culture condition. Anaerobic and enrichment media cater to these specific needs, enabling the growth of fastidious or oxygen sensitive microbes. Anaerobic media and enrichment media used for culturing aerobes which cannot grow in presence of oxygen. These media often contain reducing agent to remove oxygen and are incubated in aerobic chamber of jars. So for in easier words, enrichment media contains extra nutrients for fastidious organism, and the most common example for enriched media is blood agar and chocolate agar. Whereas enrichment whereas enrichment media sorry yeah as I said, in enriched media contains exact nutrients for fastidious organism, and the most common example of enriched media is blood agar and chocolate agar. So enriched media desire to promote the growth of particular microorganism by providing specific nutrients or condition that favor in multiplication over others. This is particularly useful for isolating microbes present in low numbers. So example is selenite broth for salmonella, and key feature is favoring specific growth. The importance of tailored nutrition for microbial studies. So in conclusion, what is the importance? Culture media are essential tools in microbiology to study microorganism in the lab. Choosing the right type of media is crucial depending on the organism you're working with and the purpose. Like the purpose can be isolation, identification, antibiotic sensitivity, and also the importance of what do you call microbial nutrition is fundamental to understanding how microbe function in natural and artificial environments. It has broad application in health, industry, agriculture, environment, and management. So both of these are very much essential uh, in our lives. It's very much essential in performing different function, and it is very much important in different fields in health, industry, agriculture, and environmental management. Without microbial nutrition and without different type of culture media, many several process won't be even have able to won't be even able to take place. So it's very important to study this, to know what is what is its importance also. But on the other hand, it's far much important to understand a culture media, and it is also very important to perform culture media in a lab. We need to do it physically in order to understand it more profoundly, and to understand what each media is teaching us, or what each media is trying to tell us. Because I think this is one of the most interesting study, and studying about each media gives you lots and lots of knowledge. It helps you understanding many things which is which was invisible for us for a longer period of time. So anybody who is interested in science, who is interested in biotechnology, who is interested in microbiology or medical laboratory techniques, if you are performing this, please perform this experiment once in your life in your lab, and you will be just surprised. These experiments, this media, we all need to do it once in the lab, and I assure you, you will enjoy it and you will learn many new things. So the importance of tailored nutrition for microbial study. The intricate world of microorganism depends precise and thoughtful approach to the cultivation, from essential micronutrients to subtle trace elements, and from general purpose brought to highly selective agas is an application of culture media essential to unlocking microbial secrets. By mastering the principle of microbial nutrition and culture media, researcher and practitioner can accurately identify, characterize, and enhance the vast potential of microbial kingdom for scientific advancement, medical breakthroughs, and industrial innovation. As I said, both of these things, a microbial nutrition is as important as a culture media, because culture media are also nutrient-rich substance or solution used in laboratories to grow, multiply, isolate, or identify microorganism. And microbial nutrients are the nutrients which is required by microorganism to perform many process, and the process includes energy production, metabolism, and cellular repair. Without this, how can any living organism perform any particular function? It won't be possible. So guys, studying about a microbial nutrition is very crucial, and along with that, understanding the culture media is far much more crucial than that. So I would suggest you to please revise it properly and understand each point properly, in order to not only just theoretically apply this knowledge, but also to physically apply this knowledge. If you are a microbiology enthusiast, if you are a biotechnology enthusiast, even if you are even if you want to become a researcher in the future, this thing is going to really help you. So guys, thank you so much for watching my video. Stay tuned for the next video, and if you like my video, please like and subscribe my channel. Thank you very much, guys. Thank you.